So I'm going to talk to you today about synthetic biology. So I guess Adam calls it wet artificial life, and I call it artificial real life. And I want to do it through some examples of what's going on in the field right now, because it's sort of a broad field, I guess, and also some of the underlying principles that at least we around MIT think are the kind of core principles for synthetic biology. And then finally, in the same vein as what I was talking about with Virgil, I want to give you guys at least some ideas on how you might get started or help out if it's something that you find interesting. Okay, so what is synthetic biology? I'll probably beat this to death, but the basic point here is we're basically adding design and construction into biological engineering. And there's been some of that to date, but the bulk of biological engineering has really been about understanding existing biological systems better, and it hasn't been a lot about building lots and lots of new systems. So synthetic biology is actively trying to make it easier to build new systems. Okay, so one of the first examples I want to talk about is a bacteriophage T7. So I know some people in the crowd here at least don't have a big bio background, so I'll try and define terms when I use them, and if I'm not doing a good job, stop me and tell me I'm not doing a good job. Okay, so bacteriophage T7, bacteriophage is basically a virus that infects bacteria. T7 infects E. coli. The reason I'm talking about it is because it's a really common, it's a very actually well-studied model organism. It's one of the first organisms that all the early molecular biology work was done on. It's, as far as organisms go, it's probably got the most known about in terms of what its genome actually consists of. It's got a very short genome, 40 kilobases. This is a schematic of that genome. Each of the genes are, well, the original 19 there are essential genes that are numbered. And they're numbered in order because the way T7 works is it basically gets drawn in, it tricks the host genome into towing it into the cell, so it gets read linearly, like a piece of code. Not all genomes are like that, but in T7 special, it's read linearly. Okay. And so the other things that are shown there, the arrows indicate promoters. What a promoter is, if you remember from your high school biology, you got DNA. Promoter binds to the DNA, it makes RNA, and then RNA gets turned into proteins. Okay, so the promoters are the sites where the polymerase binds and makes DNA. Or sorry, makes RNA. So this is about 2.5 years of a technician's work in our lab, which looking at our profiles of the RNA expression levels in minutes following infection. Okay, so at 0 minutes, you're not seeing any of the phage genes, which are numbered 1 through 18.5 there. None of the phage genes are being made yet, but then as time goes on following infection, they start to make the mRNA for these genes. Okay, this is the fit of a model that I won't get into, which is a stochastic single base resolution, very fancy model that was made in the lab. You'll notice that it gets some things right, and it kind of gets the rise on a lot of those curves correct, and it gets a lot of things wrong. Okay, why is this? Like I said, T7 is probably the most well-studied organism. Why can't we get it right? So one of the reasons is even T7, we don't know much about it, right? So all of the things marked in red there are genes of unknown function. Okay, so we have our genome, we have our code, and half of it we really don't know what it does. Okay. Secondly, there's kind of funny stuff. For instance, these are two genes. This is gene 2.8, and that's gene 3, and that's the intersection between those two genes. In T7, for one reason or another, you get overlap between genes. So this might be a form of compression because viruses, the size of their genome matters in terms of how fast they can replicate, unlike us, for instance. So maybe they're using this as a way to compress the data on the genome. But as a result, we have this overlap region. So I don't know how that works. I don't know how that affects things. Additionally, if I wanted to remove one of those genes, which is a very common way to study what it does, if I take out gene 2.8, I chop the front off of gene 3. Right? So I can't actually study 2.8 in isolation because it's integrated with gene 3. It's a pain in my ass, right? So I have no way it's not easy for me to manipulate this organism, or at least in this case. Okay, so what do we do about that? A sort of traditional approach is to make a more detailed model in T7, right? So we got to get back in the lab, we got to measure all those things that we don't know what they are, we got to figure out what those genes do, et cetera, right? A synthetic approach would be to build a new, what's that?
You'll get more PhDs that way.
Yeah, exactly, right. You certainly will give me a job, I guess. An alternative is to make a simpler organism, right? So instead of making our models more like the organisms, make our organisms more like the models, right? And that's essentially what we set out to do. So we tried to make T7.1 And that was published here. This is work from Sri Kasuri and Leon Chan in our lab. And you guys are familiar with the term refactoring, so I don't have to explain it too much. But the basic idea is here we want to keep the phage function, but we want to rewrite the genome in a way that's more amenable to our understanding and our manipulation. Okay, so how would we do that? Well, let's take that example of the overlapping region, right? So you might take those two genes and now split them up, right? So the upper line there is what I showed you previously, and then the lower line is now with those two genes. They now have separated. The one ends before the next one begins. Additionally, that colored region in the middle, D28R, D29L, are restriction sites. And what restriction sites are basically ways to cut and paste DNA. So bacteria use this as a defense system against foreign DNA, and they chop at very specific 6-base or 8-base regions. So we can put that 6-base blue region or orange region in there, and then when we add an enzyme, we can actually cut that piece of DNA out and paste a new piece of DNA in. Right? So this is how we do our manipulation most of the time, is through cutting and pasting with enzymes. This is what it looks like if you refactor the first 12,000 bases of T7. Right? So those the tall lines are individual restriction sites. So in each gene, we inserted restriction sites so that you can manipulate these genes independently. Okay. This is just to prove that it's hard and that it works. So again, this took 2 years for them to make all those little changes to refactor the genome. And then what you're seeing here is an agarose gel, and you add DNA at the top of that, what's called a well. And the DNA, basically, shorter pieces can swiggle through this gel faster than longer pieces. So a band that's further down the gel is a smaller piece of DNA. So all we're showing there is if you look at, on the left-hand one, for instance, how it's stepping up, right? You see that series of steps? That's us cutting out pieces and proving that we can take out each individual gene, okay? So then the real question is, does it work, right? So it's not refactored if it doesn't have its original function. And so on the left, what I'm showing you is a lawn of bacteria infected with phage. So a lawn means I take a bunch of bacteria, I put them on media, on a plate, and they all grow up. So I get like a thick pad of all bacteria, okay? And then each one of those dots is an individual phage particle that lands in that lawn, infects a cell, makes lots of copies, explodes that cell, it's called lysing the cell, lyses that cell, spreads phage around to all the cells around it, which then pop and spread more phage and create more phage. And so you get these circles of death, basically, right, where all the phage, all the bacteria in that circle are now dead, so it's clear. So all the area around it's still happy live bacteria waiting for their imminent phage death, okay? And so that's on the left. And then you'll notice on the right, that's our T7.1, okay? So you can see that it's capable of infecting bacteria, but the size of the circles mean how fast it's infecting, right? You know, a bigger circle means it's more infected. Okay, so it looks like it's not as effective as wild type. Maybe not surprisingly, we made a bunch of changes. But as far as we're concerned, we don't really care that much, right? So what we're much more interested in is it better to model, is it better to manipulate, and it certainly is better to manipulate. And this is, you can imagine this is the T7.2 where we knock out all the genes that we don't know what they do. We standardize the strength of things like of the components and stuff like that. So you can do lots and lots of stuff in this thing. But the general idea is use a synthetic approach, build instead of analyze. This is the first bacterial photograph. So again, this is nice, huh? This is a lawn of bacteria. This is work done down at UT Austin, actually. And so again, a lawn of bacteria, so a plate piled with bacteria, and what they look like, down in the lower left, you can see actually. You can see that they started taking pictures of things more important for their career later on. You get a nature symbol there. But what it's hard to see, but what they've done is they've taken a photoreceptor from a cyanobacteria, so a bacteria that lives in the ocean and crosses light. And they've taken it and they've hacked it out, they put it in E. coli, and they've had the output be something that basically makes the equivalent of a black dot. And so when they plate this bacteria, when they shine light on it, wherever the light hits, you express this black dot, more or less. And so you get these pictures. Great? This is kind of cool. Okay, so this is a paper about a retrovirus. Okay, and the way retroviruses work is they're RNA-based. They infect your cells. They shoot RNA in there, and that RNA gets turned into DNA, and then that DNA can get integrated into your genome. And that's how the virus replicates itself. It lives in your genome, you're the host, and then at some later point it can pop itself back out, become infectious, and spread to someone else. Okay? But it lives in your genome. Okay, that's great. But what happens if after it gets put into the genome, a mutation happens that prevents it from getting back out? right? Basically kills it, but it kills it in a state where it's still treating you as the host. So you've got this skeleton of a viral genome in your genome, right? And it doesn't work anymore. It's not going anywhere. So what they said, what happened was apparently, tens of thousands of years ago, that happened. Someone got a skeleton virus in their genome, and as their progeny bred, more and more mutations happened. And so now you could look, and a lot of people in the room would have, a particular archaic skeleton of a retrovirus in their genome. And so they could take all those copies from everybody, look at the differences between them, do some computational work, and trace back what the original genome was of the phase that first infected it. So basically, essentially using you as like a fossil, right? And so that's great, and that's kind of a normal biology approach. But what's synthetic is they turned around and built this thing, and they found it to be infectious, right? So it's really like Jurassic Park, more or less, right? Except for the virus and like your DNA, right? So they
how bad an idea was that?
maybe not the best. But
Is it dangerous? Like actually being killed?
it's not HIV apparently, but
Clearly, it didn't kill the original.
yeah.
Is this the one of the French history? I remember there was a big scandal it's like an unspoken ban on these, on sort of mimicking the endogenous sort of
Yeah, so there was a big scandal about like 1918 flu, which they they dug up a caveman basically, and or some guy, not a caveman, but somebody from 1918 who was out in the ice somewhere up north, and found like basically live viral particle, or enough that they could sequence them, and then synthesized it in order to have 1918, in order to get the sequence of the 1918 virus, which nobody knew what the sequence was. And so there's a big controversy about that. And we can talk about that later. Okay, this is interesting work. So this is basically what might be called metabolic engineering, but it's basically putting. I'll give you the backstory. So they're trying to make a thing called artemisinin, which is a malarial drug. Okay, and artemisinin is what you or I would get in the first world. It's very expensive to make. You have to grind up these trees, wormwood trees, and blah blah blah. So what they're trying to do is take the pathway out of this tree and move it into E. coli, so you can make lots of it. Okay. This isn't too different from sort of what the entire biotech industry was founded on, which was basically take a gene, we need insulin, take the gene for insulin, slap it into E. coli, make lots of insulin, profit, right? So so it's not that different, but in this case, you're looking at a much larger enterprise. You're trying to put in 15 or 20 different enzymes, and you go from a problem which was, for say insulin, put one thing in and make a lot of it, to a problem where it's like, here's a really complicated synthetic pathway. I don't need a lot of everything. I need a little bit of this, some of that. This needs to be 2 times this piece, and all this stuff. So you're talking about, basically a systems engineering problem and control theory type stuff. You're trying to, it's a chemical process. You're trying to maximize your output. So it gets a lot trickier. You start facing a lot more complicated things.
Well, you should point out that the insulin story was a lot of investment. They really messed up. Just because it's got a couple bonds, and when you do it in E. coli, it does it wrong.
Right, so it's not so simple as you go right. Yeah, fair enough. Yeah, so there's a lot of post-processing that has to go on to make it what you want. And E. coli doesn't have a lot of those enzymes. It wasn't just one thing, but the scale here is bigger. This is recently out. This is Craig Benner. It's about a month ago. They basically took genome from a mycoplasma. So mycoplasmas have about 300 kb genomes, about as small as you can get. The T7, as an example, was 40 kb. E. coli is 3 megabases. You and I are 3 gigabases, right? So so this is a very, very small bacteria. The hard part actually is getting it out and not screwing up the genome. But then they inserted it into a different species whose genome they had fried, and then divided and everything else in the first genome transplantation. Right? So what you'll see probably within the next year to 2 years is they'll completely synthesize a 300 kb genome and do this exact same thing with that, and they'll proclaim synthetic life in that the DNA was wholly synthetic, but it was booted up, right, in its organ. That's what we're really talking about. With a virus, you can just make the DNA, and that's enough, because the virus can replicate itself. It's made to just turn DNA into organisms. But for something like bacteria, you need all this extraneous stuff. The bacteria never comes out of just the DNA. It's always split off from another bacteria. It already has all that state from its parent, right? So you need something like this. You need to boot up. You can call this synthetic biology. It's glowfish now, apparently, in 3 different colors. First pet. I don't know how good an idea this is. Yeah, first GMO pet. Although they have cats too.
Yeah, they have hypoallergenic cats.
Hypoallergenic cats. So this is pattern formation work. This is done out of Ron Weiss's lab at Princeton. So again, there's a lot of bacteria, so I think I was talking about before. On the left, you can see there's a little circle tab there that gives off a chemical. And then the circuit that's in these is basically a bandpass filter. And so within a certain range of that chemical, you're going to get a diffusion gradient away from the source, and the cells will either grow green or red depending on what gradient of the chemical they detect. Okay. And so you can use it to make circles and hearts and all kinds of stuff. But what underlies that particular project is near and dear to me is basically genetically encoded logic, right? So at the moment we are extraordinarily bad at doing any kind of logic in cells, and this is basically the cutting edge. And what you're looking at is an AND gate, a ring oscillator, and a bistable switch, right? So this is what we can do. And on top of that, it's not only that this is what we can do. Unlike in a say electrical system where you have physical separation, where if I make one AND gate, I can make then 100 of them and put them all together. If I make one AND gate here, I don't have physical separation. I can't actually put it together with its twin in the cell because there's crosstalk between all the genes. It's all just a bag of cell, it's a bag of chemicals. These things can spread from one AND gate to the next. So really, I literally only have one AND gate, one ring oscillator, and one bistable switch here. Okay, right? So this is kind of where we're at. And I'll talk about what that means with respect to the principles. So I hope I've gotten across the idea of synthesis as an approach, of building things rather than sort of, I don't know, just trying to drill down and understand in more detail. A second and third thing are sort of characterization and standards, and then abstraction. And I'll talk about each of those. So why do we need that stuff? Why do we need standards and abstraction? So as I gave an example, this is the ring oscillator, right? So this took 10. This took about 2 years to make by probably the best biochemists in the country, right? It's extraordinarily difficult to do this stuff, okay? And additionally, it's not easy to use this as a subcomponent of a larger system, okay? So I'll talk about that. Okay. This is what we're working with. If you're a genetic engineer, this is your machine code, right? So you're coding in BASIC. What you'd really like to be doing is treating that as a part. Okay, so I don't really want to know ATTA, TGC. I want to know B0015. I want to know what it does. It's a transcriptional terminator. It stops polymerases. I want to know its efficiency, how it operates, and things like that. I call that a part. I want to combine a bunch of devices together. I won't go into great detail, but this is what we would call a genetically encoded inverter. And you know what it's supposed to look like in terms of its input and output. Again, I'd like to put a black box around it. I'd like to give it a number, right? And this is going to and this will let me, now if I want to make a ring oscillator, I could take 3 of these things and hopefully put them together and have it actually work, if this is an inverter, right? Okay, so we're starting to collect these things, these parts, right? So there's a website which I encourage you to check out, parts.mit.edu, and you can look at parts by different types, right? So there's drivers and line sites, motors, inverters, all kinds of different stuff, right? So you can go here, and I passed around a little square thing, DNA, I don't know what that is. There it is. That's 1/4 of all the parts in the registry are in that right now. So there's dry DNA down here. So don't lose it. No, it's good. You can copy it. Yeah, it's not worth it. Alright. Okay, so measurement. Here's an example of something which I think I bring up because I think it's kind of relevant to the A-Life crowd here. Okay, so this is a device we built to measure, say, inverters, for instance. So we have green fluorescent protein which looks green to measure the input, red fluorescent protein which looks red to measure the output, right? This is an inverter from the registry. It's QO4401. It doesn't look like an inverter to me. It's a flat line, right? You see for low input I get low output. That's not right. So how do I fix this thing? I tried to fix it by hand. I made some changes to the sequence as best I could predict, and I just broke it worse. I made it the opposite thing. It was always on. So instead, one thing that biologists have been doing well relative to other engineering fields for a while is using directed evolution, right? So in this case, we made a library of 100,000 variants of an inverter. We slapped them all into that device, and then we collected the ones doing what we wanted using a high throughput sorting machine, right? So I say, okay, give it a low input, collect a high output. Give it a high output, collect a low input, and then I can get something that looks more like an inverter. Okay, so we're using, so in my opinion, the A and life stuff, biologists are actually relatively ahead of the curve in terms of engineers in terms of using evolution. they're kind of way behind the curve in terms of using abstraction and standards and things like that. But we get it. It's great. This is a data sheet. So this is probably the best example of a first generation data sheet for a biological device. It takes in a small molecule. In fact, it takes in a small molecule from those pattern forming things I was talking about. And you can see all kinds of different stuff about it. How fast it responds, what the transfer curve looks like, all this stuff. Okay, so if you guys think this stuff is cool, what can you do about it? Is there any way you can participate? So one thing is the iGEM competition. So this is the International Genetically Engineered Machines competition, iGEM, okay? And what it is it started off with a class at MIT like 4 or 5 years ago. Then it had 5 schools in it, and this summer it has, I think, like 70 schools in it, and from all over the place, China, Japan. Basically, the competition is get together a group of undergrads and some advisors, spend 4 months, and come back with the coolest genetically engineered machine. It just has to be a biological system. That's kind of the extent of it. And if you make any parts, you got to give them back to the registry, right? You got to share your parts, and additionally, you get wherever that thing went. You get 4 of those things, plates of DNA. So you get every part that we have first day of the summer. You got them, do what you want with them, make stuff. Okay, so that's the competition. I personally think that if anybody here was interested in participating in this, you could easily get in touch with the Harvard, MIT, BU team, or whatever, and tell them you want to hang out for the summer and help, and they'd probably take your help. Free charge. So that's one thing. I'll talk a little bit, real briefly, just to give you an idea of, because one of the problems with this stuff is that there's still relatively high barriers around getting involved, right? You need some relatively expensive lab equipment. You need a lot of sort of these annoying protocol techniques where little details matter. It's easy to screw stuff up, right? So there's a lot of latent knowledge that is still a big barrier for hobbyists, people just getting involved in this and more people using it. So what I want to give as an example, this is a project which was done again by 5 undergrads with a couple of grad student advisors in a period of 4 months. The goal was to make, this is the Eau de Coli project, sure. It was to make bacteria which normally smells like crap, to smell like mint during one phase of its growth and like bananas during later phases. So how do you do this? Well, it turns out you grab 4 or 6 different enzymes from petunias, yeast, and all the different organisms, and you bring them in, and you put them under different control. So we use some existing parts from the registry. And basically, they built a system which probably had about 20 parts, 20 different construction steps, which is really, I try to give you the scale because I think people don't appreciate it, but that's a lot of work in normal terms. And they were able to do that in about 4 months because when parts go in the registry, I didn't talk about this, they have basically a construction standard. So they have to go in a certain way so they're easy to put together. So it means that a group who's using parts from the registry can build stuff relatively quickly and produce something like this, which actually ended up working, in 4 or 5 months with pretty much no previous lab experience, which is a big deal. So that's why I think, if people want to plug in, most of these iGEM teams don't have a lot. I mean, these are freshmen and a couple sophomores. And this is cutting edge metabolic engineering. Another way to plug in is CAD tools. This is the cutting edge of our horrible CAD tools in biology. This is Vector NTI from Vitrogen. It's pretty good at marking up sequences. This is what this is what that is. This is Gene Designer. It's the best tool for putting pieces of, I want to add this and this together. So it's great for putting 2 things together and saying, well, this is what they equal when they're together. What it doesn't do for me is say, oh, you want to put part A and part B together? That's a bad idea. Those aren't going to work together. There's no compiling going on here, right? It's not nothing smart. No one knows. It doesn't let you know if there's a mistake or anything else. So you're pretty much as good. I mean, T7.1, the editor, the CAD for that was like TextPad or something, right? It was done in Word. Garage Biotech. So this stuff, again, I think we're a ways away from this happening. I passed around Make Magazine. Passed around the DNA Wizard kit. I like the DNA Explorer kit, which is pretty cool. So Meg talks about how you can make a gel electrophoresis chamber. That comes with a gel electrophoresis chamber and a bunch of other stuff. It's pretty cool. It's got a centrifuge. $55.
So how soon does Homeland Security show up if you order one of those?
I was pretty impressed by it. It's won a whole bunch of awards. It's a really neat kit. Anyway, so.
It's not chemistry, biology.
What's that? Yeah, Homeland Security is worried about chemistry. That's right, yeah. If you got a little more money, you'd probably get yourself a synthesizer on eBay. There's a whole bunch of good resources online. I'll just point you to briefly. Again, the make stuff. There's an interesting article published in 2600, like 4 or 5 years ago, by some anonymous professor. That's pretty interesting about how you might get started doing biology. The main recommendation is to take a class. That Journal of Visualized Experiments is pretty good because you actually get videos of things. It turns out that having somebody show you how to do something ends up helping a lot doing this. So I like that idea. I'm involved with openwetware.org, which is basically trying to get labs to kind of exchange their information prior to publishing it, so get more out there, exchange the tacit knowledge that people don't normally trade. Protocols Online is an excellent forum. You want to ask them questions there. BioForge is supposed to be like a source forge for biology. It hasn't really taken off. Although it's a great thing. And BIOS, which is the organization behind it, if you're interested in like open source biology, you should check them out. They're really doing some good stuff. The Registry of Parts, and then LabX, which is just a place you can buy used equipment if that's your thing. And that's it. I stole a lot of slides from my boss. These are people who helped me out from the lab. NSF pays the bills. That's it. So, thank you. Questions? Thoughts?
Just to kind of assemble this into like a big picture goal. When you talk about oscillators and inverters, you sort of set up a model where maybe you're trying to use all this to create a better computer. And I think some funding has gone in that direction, but all of the output has really just been biological results. Make a plant that smells like 2 different things, or make a plant that goes through a prototype. So what is oscillator? What's the end run now? Why are we doing all this?
So I'll give you my personal opinion. So I think the computing angle of the like biology has a better replacement for silicon made career. So and I don't think that's the goal of most people. Right? We're not going to compete with silicon for speed. That up fast. But what would matter is small amounts of memory and logic applied to biological systems. Well, right now there's right. So even if we had very simple levels of control for things like studying, let's say, you know, we'd like to be able to count to 10 in cells. You know, track that. That would help us study aging, right? You know, put that in yeast and know how many things. Right? The normal process right now is to take the yeast. Every time a baby yeast comes off, it's a scar. So you put the look on the microscope and count the scar to find out how many times that yeast is alive. Now you certainly can't do that in my field, but if you had something like a counter, like an output you after 8 divisions, sort these things all kinds of things. So I think the initial application would be in tools to help people do research better, at least as far as one. I think you're absolutely right. I don't think there's big. I don't think there's big.
How clear was the combination? I had the impression that you can't just take 2 or 3 quarters and hook them up together and expect that everything will work the same as if they were separate parts.
Yes.
Can you say something about some of the effects for changes? You're not losing money changing. What are the things that help you determine what will be the effect of throwing something in?
Yeah, so that's very much so we call it composability. Big heavier research rights. We've got these parts, even if we have them and we have them all well characterized, when we put them together, are they actually going to work as predicted in series? Like, you know, as I said, it's a mixed bag thing, right? There's certainly going to be crosstalk across different devices, right? Levels of expression that have to match each other. There's going to be figuring out which is load on the cell. So the cell has a fixed amount of resources to run your device and to run its own survival. You have some really devices cross heavily on the cell, it's going to break. We've actually seen a lot of that. We've actually seen, if you put a really big drain device in there, basically the thing will fall around it and break as fast as it can, so people will take off and that's the end of that. Right? So yeah, I think you're right, and it's an open area research. I don't know. We have to, there's certain things we can do to try and design parts, at least so that theoretically they go together. So like for instance that inverter I showed you is designed specifically so that it takes the input and has an output delivery. So we have a common signal carrier in that. Depending on the amount of polymerase that come in on the DNA that enter this black box, high in or low out on the other side. So you at least have a common signal carrier. That wasn't always the case. Originally they were designed take in one thing without something else, and you combine them together without a ton of things. So,
Do you use any analysis like kinetics or thermodynamics to try to
yeah, absolutely. So you can measure, you can use, I mean, a lot of these things are well studied. So for instance, in burger is a repressor, right? The repressor is basically binds to the DNA, they bind it from the other side, and prevent polymerization, right? And that's well studied. People know how to measure the kinetics of the repressor. So that can go back into a model, and you can use that model then to decide, okay, well, how well is this device going to repress that device based on the kinetics, right? That's certainly true. You know, in practice, I think the first thing that'll happen is we'll be great. We have a lot of things that exist that are easy to combine. So even if you don't have great models, which is most frequently the case, try a lot of stuff with it, right? Because like right now, we don't have good models, and it takes us a long time to try stuff. It's horrible time. One cycle is really slow, and it's slow, and on top of that, it's almost like a fail. There are models.
Are you trying to make simulators that would
Yeah, so there are, yeah. So there's work being done to make a simulator so that when you make things in the parts out of the registry, each part has like a model attached to it. So the registry of standard biological models go along with our registry of standard biological parts. So that when you put the two parts, their models will be combined to predict how it's going to look. With a great result. I mean, modeling has gone a long way in biology. It's still, like you saw in T7. Yeah, that's a great model and a really well studied one. The model's simple, but as we get more complex.
Further, did you go back and figure out what changes the organism made?
Yeah, so it was actually a change that was very similar to the change that I had made when I was trying to fix it myself. But it hit it in the right spot, you know, the golden box. You know, it got warm and I just went too high. So it was just the strength of the right So I mean, I think vector evolution is a great tool, even though we're behind a sort of standards idea where we want to make parts that work together. Vector evolution is a great tool to make those parts in the first place. Right? So that's how we do it.
Say you wanted to take directed evolution a step further, and how would you actually rig up an environment to confidently select for your inversion? So you spread a bunch of gunk down a line, pick up the things that seem to be going high with low, low with high, and smash them together and do it again.
Yeah.
Could you do that in one step?
Yeah, so so you can imagine attaching it set up. So I use fluorescent proteins as my outputs and inputs for a particular reason, but you can imagine doing a similar thing with antibiotic resistance genes, so that you express a certain amount of antibiotic as the output, and then based on how much antibiotic is present in the plate, something that's expressing high lives. So you're actually, instead of, what I do is take a protein from a machine and run them through and collect what I want. You just plate these out, and only the ones that work live. So that you can stream more that way, like billions and billions, and two of them live.
It seems like that thing is really high throughput is the necessity for technicians to go over and cut a piece of the gel.
Well, actually, it turns out that that's pretty high throughput the way I can do it. So with those flow machines, you can do thousands of cells a second right through it. So it's pretty cool. I mean, I specifically chose not to do the antibiotic resistance because I wanted the fluorescence is more sensitive. So I can choose, I want it this high, not that high. But antibiotics is not.
Use 2 years for the work. T7.1.
Yeah.
So where did the time go?
Yeah, manipulating data. So you want to go in, you want to make, whatever it was, a couple of 100 changes. You got to go in there, and if I want to make one change in the genome, a single base pair, I'll make that. If I want to make, move the whole section, that also, I mean, basically, anything you kind of want to do takes a quarter a day or two to make that change. So you can parallelize it in some respects, you did, but it's just slow. Basically, that entire thing is just built, but to put it in perspective, direct synthesis, protein synthesis, eBay over there, that's finally getting cheaper. So for T7.2, we're just going to directly synthesize the thing for $10,000 in 2 weeks, right? Versus 2 years. So we're at the point now where pretty quickly, this is another thing that's driving all this. Pretty quickly, our ability to make stuff is going to outstrip our ability to design. Right? So we're going to soon be able to make things in the complexity of like here. We don't have design systems. Our parts aren't good enough. Right? So you're trying to, somebody needs to write programming. Well, that stuff has to happen. Yeah, I mean, I think the best way to automate is going to be like for that particular case, which is manipulating DNA. It's going to be like synthesis. So these like there's a lot of those companies now that are all over the world basically working to make it's bigger and bigger. And that was like the Benner thing. Like, they're going to synthesize 300 kb genome in 2 years, you know. And so that's going to be the scale we're at.
The other thing is you seem to always be assuming linear transcription from DNA to RNA to protein, whereas eukaryotes anyway seem to have introns, so
I'm glazing over seas of complexity here. Right? Yes, absolutely. Right. So there's sorry. Okay. Yeah. So I mean there's yes. Right. So there's all kinds of useful stuff that particularly eukaryotes, even prokaryotes, that may be more complicated. And eventually, the idea is to extract all that stuff away. You know, this is how we want introns to look like. This is what standard protons look like. So that the start site on all, even something so like a motor. If you look at the motors in the registry, they all have different start engines, so it really matters. Right? So like we have a long way to go. Just applying standard. A lot of work.
Broad estimate.
Yeah.
In terms of how far we are from bioshock, you know, now I can make finger, fire with my finger.
Plasmids over there.
Yeah. Is that 1,000 years, 100 years, or 10 years away?
I think it's a really simple question. I think there's going to be some of them are going to be able to do it. I'm sure we'll be able to. Well, I mean, we already could manipulate. You might manipulate light all the time. But to answer your question, I don't know.
To get it under control enough so this could be possible,
Yeah.
and if it's stable enough that I would be willing to do it and not like risk death, you know,
Right.
1 in 10 million, but not 1 in 100,000.
Right.
My lifetime?
I don't know. Maybe if we get the aging stuff right.
I've wondered is what happens when the extreme body modifications happen genetically? You know, you've got like you know people like you get on the subway and there's like a guy on there with like glowing eyes and yak hair and tail and horns. Freaking awesome.
Yeah, I think I'll leave it at that.